Hello everyone, and welcome back to AP Bio with AP Playground. We finished Unit 1, and now it's time to review Unit 1, Chemistry of Life. The main things you want to know within Unit 1 include the structure and properties of water, carbon being the center of life, basically anything and everything about the four major macromolecules, the structures, and functions, and that includes the difference between DNA and RNA, and how they're put together and come apart. Before we really get into the content, we've got to cover some simple chemistry, so let's talk about bonds. First of all, we have covalent bonds, sharing of electrons within the valence shell. So for instance, oxygen, which has a valence of 6, would have 6 electrons in its valence shell. And a valence shell can hold up to 8 electrons, meaning it can fit 2 more electrons. And then a hydrogen atom right here, could come in with its singular electron. And now all of a sudden, the hydrogen and oxygen are sharing that electron, and they formed a covalent bond, which is really hard to break. Ionic bonds are fairly simple. It's just if you've got a positive ion and a negative ion, so a cation and an anion, they're going to attract and they're going to come together. Next up, we've got van der Waals forces or London dispersion forces. This is a close interaction of electrons and they form weak bonds. So basically, if you've got an atom here, we'll call this, it doesn't have to be any specific atom, and an atom here, it's got electrons flying all around it in the electron cloud. We've got electrons here, electrons flying all around here. So there's a chance that within this atom, this is like a zoom up image, there's a chance that more electrons are going to somehow end up on this side randomly through their movement of bumping around than there are on this side. And then the same thing can happen with the other atom, where there will be more electrons on this side flying around and less on this side. So all of a sudden, we've got a really slightly negative charge here and a really slightly positive charge here, and the same thing here. And as we know, opposites attract, so through this very slightly negative and very slightly positive charge, this partial charge, we're going to be able to get a very weak attraction between the two atoms. Now the big one here, especially when talking about water, are hydrogen bonds. So the polarity of water causes either side of the molecule to have partial charges. So if we take a look at a molecule of water, right here, a molecule of water, water is made up of one oxygen atom and two hydrogen atoms, and these are bound together covalently. So they're sharing the electrons. But oxygen is electronegative. It's a much larger element than hydrogen, and if you look on the periodic table, it's way up in this section. So if you go up and right, you're going to get electronegativity. So oxygen is really electronegative, so it's hogging the electrons from the hydrogen and trying to bring it over to its side. So what's going to happen as a result is we've got a partial negative charge here with the oxygen because all the electrons are hogging around it, and a partial positive charge on the side of the hydrogen. And what happens now is we've got a partial positive and a partial negative charge, so multiple hydrogen atoms can connect between the partial positive and partial negative charges. And this is how you form what's called a hydrogen bond. Okay, let's talk about the properties of water, starting with cohesion. That's when water is attracted to itself. So, for instance, we've got a couple of water molecules here that are going to be attracted to each other through hydrogen bonds, and this is going to form what is called surface tension. So the oxygen attract to the hydrogen. So because we have this water bound, if you have a river or a lake, the top of the river or lake is going to have more or less of a film over it because all the water is bound to each other. And this allows creatures such as a water strider to walk on top of water because of this small film. Now, adhesion is the attraction of unlike molecules, water, for example, water to glass. So if we have a beaker here, a glass beaker, and we're just going to fill it up with water, it's going to make a sort of curve like this. Because it's attracted to the glass here, it's going to creep up the edges of the beaker. And this also causes transpiration. So transpiration is evaporation from a tree. So if we have a tree, here, and then we give it some leaves, traveling up the xylem tube is going to be water from the ground. It's going to be pulled up the roots, and then it'll be evaporated. And this is powered by the sun. But the way water is able to travel against gravity is as the water holds onto the xylem tube, as it uses its adhesion to hold on to the xylem tube, it's then pulling up the rest of the water molecules through cohesion. And that way it's able to defy gravity and evaporate. Now, water also has a high specific heat, which means it resists temperature change. So if you've got a coastal city, the coastal city will have a much more moderate climate because the water won't heat up as much and it'll take longer to cool down. And this is the same with high heat of vaporization. When you're sweating, the water is going to take a while to evaporate off of your body. So once it does evaporate, all the heat is going to go with it. Now, water also has lower density as a solid. Ice floats. If you ever had a cold drink on a warm day, the ice is going to float in the cup. And this is pretty unusual because most solids are more dense than the liquid form. And this is going to protect ponds because when ice forms over a lake or a pond in the winter, their life can still thrive below this layer of ice, which is only at the top because it's floating. And lastly, water is a nearly universal solvent and can dissolve any charged substances, or those that are hydrophilic or polar. It cannot dissolve hydrophobic or non-polar substances. Moving on to 1.2, now we're talking about carbon. So all major macromolecules of life contain carbon. Now, what is a macromolecule? Macromolecule is just a large macromolecule, and there are 4 of these that are necessary for life. But speaking of carbon, carbon allows for many bonds to be formed because it has a valence of 4. And with a valence of 4, that means it's got another 4 spaces left in its outermost electron shell, so it can bond to 4 other elements, allowing large structures to be formed. And compounds with carbon are considered organic compounds. Now, there's a rule of Chomp, which basically shows us what the different macromolecules are made out of. So the 4 major macromolecules are carbohydrates, lipids, nucleic acids, and proteins, and you can tell what elements each one is made out of using the rule of Chomp. So carbohydrates are made out of CHO, and so are lipids. Nucleic acids are made out of CHONP, and proteins are made out of CHON. And what this means is that they're made out of certain essential elements necessary for life. Carbohydrates made out of carbon, hydrogen, and oxygen. Lipids made out of carbon, hydrogen, and oxygen. Nucleic acids also including nitrogen and phosphorus, and proteins also including nitrogen. So if you're ever in a pickle and you need to remember what constitutes the macromolecules, use your CHOMP. CHO, CHO, CHON, CHOMP. Now, besides carbon backbones, which make up the molecules and macromolecules, there are also functional groups which attach to the side, which give different properties, such as polarity, making it acidic or basic, changing the pH, or it could be nonpolar. So there are just a few that you need to know for biology, starting off with a hydroxyl group. This is just an oxygen attached to a hydrogen. And because it's bonded, we've got the electronegative oxygen. It's going to be polar molecule because we've got those partial charges. And what this R means is just the rest of the molecule. Now, methyl group is nonpolar. It's just carbon attached to some hydrogens. Next up, we have a carbonyl group, and this is polar because we've got an oxygen. And there are 2 types of carbonyl groups, aldehydes and ketones. A ketone is what's listed here. You've got surrounding compounds, so it's just in the middle. A ketone fits in the middle of the whole, whereas an aldehyde would have the R compound, the double bound oxygen to the carbon, and then ending with a hydrogen right here. That would be an aldehyde. Aldehyde died at the end. These are present at the end of whichever compound you have here. And they're again polar because there is that oxygen which can become electronegative and form a bond with hydrogen. Now, carboxyl group is very similar to an aldehyde. There's just a hydroxyl group instead of just a hydrogen here. And besides being charged, it can also release a proton, meaning it can be considered acidic. because what happens here is that the proton off of this hydroxyl group can be released into the environment, leaving an oxygen with just an electron attached, and the proton is now in the environment, meaning this is an acidic molecule. Now, an amino group is a nitrogen attached to two hydrogens, and because it's got a larger valence, it can accept another hydrogen, it can accept a proton from the environment, meaning it's basic. Because when you're adding protons to the environment, you are lowering the pH, it becomes acidic, and when you're removing protons from the environment, the pH goes up, it becomes basic. Now, a phosphate group is a phosphate attached to four oxygens, one of them being a double bound, and again, through these hydroxyl groups, it can release protons, meaning it is acidic. And lastly, sulfhydryl group is just a sulfur attached to a hydrogen atom. Next up on 1.3, there are monomers versus polymers, meaning subunits versus the whole. Think about Legos. Individual Lego blocks would be the monomer, while Lego blocks put together to form a structure becomes the polymer. And every macromolecule is made up of some sort of monomer, except for lipids, which form polymers. So some monomers of carbohydrates and proteins form polymers through what's called dehydration synthesis. And dehydration meaning you're losing water, and synthesis meaning means that you're taking away water to combine two monomers. So for instance, over here we've got two glucose molecules. We're removing water by taking away a hydroxyl group and a hydrogen atom from the two molecules of glucose to form water as a byproduct, and using that to connect the two glucose molecules through a covalent bond by the oxygen, forming the maltose molecule. So these two monomers are forming the polymer. And the bonds can be broken through hydrolysis, which is the reverse. Hydro meaning water and lysis meaning break. So when you again add that water back to your newly formed molecule, if we add the water back to maltose, it'll then become two glucose molecules because we've added those elements back which the molecules need. So dehydration synthesis and hydrolysis are opposites and they are reversible, but this is how carbohydrates and proteins form polymers. Now on to carbohydrates. From now on, we're not going to talk about specific subunits, but just the macromolecules as a whole. So the polymer of a carbohydrate is a monosaccharide, meaning one sugar, while the polymer is a polysaccharide, multi-sugar. And these polysaccharides can either be ringed or branched. You'll often see glucose as a ring with 6 carbons, but you could also have branching out chains of monosaccharides. So they're connected by dehydration synthesis through glycosidic linkages. That's the name of the bond that is formed between two monosaccharides in a carbohydrate. They have isomers, which can either be structural isomers or enantiomers. Structural isomers are just bonds in different places. So if we look at the glucose and the fructose at the top right, we'll see that there's this carbonyl group that's in a different place. Here it's basically a ketone group, while in the glucose it's an aldehyde. So there's the chemical formula is the same exact formula, C6H12O6. However, there is a difference in the structure of the bonds. And it's important to also mention that all carbohydrates are composed out of CHO, carbon, hydrogen, and oxygen, and there's usually a 1-to-1 ratio. So like in glucose, C6H12O6. Enantiomers are simply just mirror images, which we can see between glucose and galactose. More or less everything is the same except for this hydroxyl group. So even a difference in the positioning of the ring can also affect the carbohydrate, giving it different properties. So if we look down here, there's an alpha versus beta glucose. And when you have alpha glucoses, then you're getting yourself to starch. But when you use beta glucoses, you become cellulose. And it's not that there are any different chemical formulas, it's simply just the way the positioning of the rings and how they're connected affect the carbohydrate. So in the alpha rings, you can see that there's always a bond on the oxygen, it's always going to be down here. However, with beta glucose, every second glucose is flipped around. So there's this different type of structure being formed, and that affects it to the point where we're getting a different thing, even though a different compound, even though the chemical formula is the exact same, and all of the monosaccharides, all of the monomers are the exact same. So carbohydrates are used for energy and structure. Some energy storing molecules are starch and glycogen, starch in plants, glycogen in humans, and structure, cellulose and chitin. So cellulose is primarily found in plant cell walls, and it's what makes plants so rigid, and chitin is found in the exoskeleton of lobsters and animals like that. So the important thing to think about is, how does appearance affect activity? How does form follow function? So when you have a branched carbohydrate versus a ring carbohydrate, you've got some different things, because a branched one, you can easily break it off to gain the energy, whereas a ring carbohydrate could be possibly more solid to give more structural support. So depending which function we're using, energy or structure, there's going to be a different structure of that carbohydrate. So if we look down here, these are cellulose molecular chains. We can look, they're chains of carbohydrates, and they're even bonded by hydrogen bonds, and they're formed in these sheets, which make them very strong and very good for providing structural support for the cell wall. Whereas if we were looking for glycogen or starch, these would be branched so that you can more easily break off carbohydrates and gain energy from the breaking of the bonds. Okay, moving on. Now we're moving on to lipids, and these are also CHO. However, they're not really considered macromolecules because they don't have monomers that make them up. But because they're so important to life and they are so large, they are a large molecule, they're considered to be macromolecules, in not by definition, but just by name. And they are nonpolar, meaning they're not going to be interacting with water. So there are a couple of different kinds. First of all, triglycerides, which you might see in the name, is going to have 3 fatty acid hydrocarbon chains, triglyceride, and there's also a glycerol backbone. So the interesting thing here about these hydrocarbon chains is they can be saturated or unsaturated. So a saturated hydrocarbon chain would be such as one that appears here in this triglyceride. Every single carbon has hydrogen in all of its free spaces, all of its free bonding spaces. Everything is completely filled up. However, an unsaturated hydrocarbon chain might be missing some of the hydrogens, so as a result, there's a double bond formed between 2 carbon atoms. And this is going to cause what's called a kink. Because of the double bond, there's going to be a bit of a change. Instead of just being a straight line, it's going to be a slightly bent structure. And this is going to then affect the function. Generally, when you're looking at saturated things, they're going to be solid at room temperature, but unsaturated hydrocarbons are going to be liquid at room temperature. Because of these kinks, there's more separation between the molecules. So there are also steroids, which are 4 carbon rings, and they're generally found in hormones and cholesterol, or these are examples of steroids. And then lastly, phospholipids, which you'll see in the cell membrane, and these consist of a phosphate group, which is polar, so it has this attraction to water, plus 2 fatty acid chains, which are obviously non-polar because they're fatty acid and they're lipids. And this is going to create the cell membrane structure, which we'll go into next unit. So lipids store energy long term, and that's the reason why there's such long branches of just hydrocarbons. So again, this is appearance affecting activity. Their structure of being really long and hard to break apart is good for their function of storing up energy long term, because even though they have a lot of energy, it takes energy to break that up. So they're going to be used after all of the short-term energy in carbohydrates is broken down. And they make up membranes and obviously act as hormones, which is what we saw with steroids. So there's another kind of fat, which is trans fat, which is an unsaturated hydrocarbon, which is then saturated unnaturally, and it's used to be in a lot of junk food, but it's very unhealthy and dangerous, so it's been basically banned more or less. And that's it for lipids. Okay, now we're on to the big ones. We're on to amino acids. And our formula for the majority of these is going to be CHON. So carbon, hydrogen, and oxygen, but also containing nitrogen. So proteins are made out of amino acids, which all contain the same backbone but have different side groups. So if we look down here at this amino acid structure, every single amino acid is going to have a central carbon, a hydrogen atom, a carboxyl group on one end, and an amino group on the other end. And this amino group is what's going to be that N in the CHON. And each one is also going to have a different side chain that extends, that protrudes from it, which would give the amino acid different properties, because it could be polar, non-polar, it could be acidic or basic. And there are about 20 of these essential ones that you might see in this course, but it's unnecessary to memorize them. You just have to be able to recognize, are we looking at something that's going to give it a property that is acidic, that's going to give it a basic property, polar or non-polar. And these amino acids are connected through peptide bonds, which through dehydration synthesis, which forms a polypeptide. Now, proteins are used in basically everything. They're used in building and repairing the body, for gene expression, for movement, for enzymatic activity, for defense, like antibodies. So there is a bunch of uses for proteins in the body, and they're one of the, I don't know, you could argue that they're the most essential macromolecule, even though all of them are essential. And how does appearance affect activity? You've got a change in the structure is going to affect the shape of the protein, which could affect the function. So if we look over here on the right, we've got a bunch of different amino acids connected to each other. We can see all of these are side chains protruding, and each one's got different properties. And all of this is going to contribute to the shape of the final shape of the protein, which is then going to determine what it can do. So now we're going to talk about how we can determine this shape of the protein through the different stages of protein folding. So we established that proteins are made out of separate amino acids, but each protein is different because of the different properties of the amino acids in determining the shape. So there's the primary structure, which is just the sequence of amino acids. Then secondary, which is hydrogen bonding between the amino acid backbone, which can either result in an alpha helix shape or a beta pleated sheet. So if we look down here at the bottom left, here's our primary structure. We've just got all of the different amino acids in a row. But then because of the amino acid backbone, because of the carboxyl group, we are able to do hydrogen bonding. And this can result in, here it's shown the alpha helix shape, but it could also create a beta pleated shape, which is more like a sheet. Then in tertiary structure, this was just the amino acid backbone folding. Now we have to take into account all of the side chains. And these can lead to ionic bonds, which are obviously between a positive and negative ion. Disulfide bridges between two sulfhydryl groups. Hydrogen bonds, which we've already seen, and also hydrophobic interactions, also known as van der Waal or London dispersion forces. And what this means is because there are some amino acid groups, some amino acids that are going to be very nonpolar. They're going to want to be in the very interior of the protein, whereas some will be polar. They want to be in the exterior. They want to be they want to be surrounded by the water or whatever other solution is outside the protein. So because of such folding in the interior, the amino acids are going to be so close to each other that there's a chance that even with the random movement of electrons, there might be a slightly positive side to the amino acid and a slightly negative side. Because of the electrons moving around randomly, there's a possible dispersion of electrons that they're slightly to one side or another, and this can create a really weak and small attraction force. The hydrophobic side chains want to curl inwards away from the aqueous solution. So when we get this really small attraction force, it's called a hydrophobic interaction or a van der Waal force. And it's literally just due to the random movement of electrons and because those nonpolar amino acids are in such close proximity to each other. And this gives us our tertiary structure, the polypeptide chain, all folded up because of the side chains. Lastly, there's quaternary structure, and this doesn't go for every single protein, but some proteins have multiple polypeptide chains. And once they attach to each other, we get quaternary structure. And they can attach to each other in the same way as in tertiary structure: ionic bonds, disulfide bridges, hydrogen bonds, and hydrophobic interactions. Now, when the bonds in secondary and tertiary, and obviously quaternary as well, structure break due to a change in pH, salinity, or temperature, the protein becomes denatured. These bonds can be broken apart because they're not covalent, they're not very strong. They hold the protein in place, but changes in the environment can result in breakage of the bonds. However, primary structure cannot be denatured because it's just the sequence of amino acids combined through peptide bonds, which are covalent, meaning you can completely unfold the protein, it can be denatured, but the entire protein will not break apart because you will still have your primary structure, your sequence of amino acids, which will remain intact. Last group of macromolecules, we're going to talk about nucleic acids. So we've got DNA and RNA, which are the two different kinds. We're going to talk about DNA here, RNA on the next slide. And they are formed from nucleotides. That's the monomer, and nucleotides bonded together covalently form strands of either DNA or an RNA. And all nucleotides contain a 5-carbon sugar called pentose, and we can count these off as we look over here on the right. We've got a 5-carbon sugar. This is an oxygen molecule, so the carbons, you can start counting the carbon from here. So this is carbon 1, 2, 3, 4, and 5. A phosphate group, which is going to be seen right up here, and then lastly a nitrogenous base: adenine, cytosine, guanine, thymine, and uracil, and they are paired A to T, adenine, thymine, and C to G, cytosine to guanine. Adenine also pairs with uracil, but that's only in RNA. And a good way to remember this is apple tree, car garage. And adenine and guanine are 2-ring, they are purines, while cytosine and thymine are 1-ring, they are pyrimidines. And strands of DNA run anti-parallel, meaning even though they are parallel, they run in opposite directions, and each one from the 5' carbon to the 3' carbon. And meaning we've already counted the carbons. We know that right here is the 5' carbon and right down here is the 3' carbon. This strand is going to run in this direction. However, the 5' carbon for this other strand is here and 3' is there. We're running in that direction. So they're running in opposite directions, but they are still parallel. And we mentioned that nucleotides are bonded covalently. So they're bonded covalently between the phosphate groups and the 5-carbon sugar. We can see that they're forming this strand, and all of these bonds here are going to be covalent. The bonds between, this is a covalent bond, there's a covalent bond, there's a covalent bond, and same thing on the other side. However, the nitrogenous bases that combine two strands are not covalently bonded. These are going to be bound with hydrogen bonds. And as you can see, between cytosine and guanine, there are 3 hydrogen bonds, whereas between adenine and thymine, there are only 2. Which means the bond between cytosine and guanine is slightly stronger. So while the individual strands are going to be bound covalently, strands bound to each other are always through hydrogen bonds. And DNA's purpose is to store and express genes. And we can think about it this way. How does the appearance affect activity? DNA has 2 strands, meaning it's easy to copy one, because we can always create a template and use one as a template and the other one just gets copied. So this is how DNA works. Now moving on to RNA. Here we go. To finish up, let's talk about RNA and its differences from DNA. RNA is single-stranded compared to DNA's 2 strands. The nitrogenous bases are adenine, guanine, cytosine, and uracil instead of thymine. So adenine pairs with uracil if you combine 2 RNA strands. And there is a ribose sugar instead of a deoxyribose sugar. A deoxyribose sugar in DNA, which is deoxyribonucleic acid, is not going to have an oxygen. It's deoxygenated. So if we look down here on the right, ribose is going to have a hydroxyl group in its 5-carbon sugar. It's going to have an oxygen and a hydrogen. However, the deoxyribose will not have the oxygen and only a hydrogen. So what is the purpose of RNA? RNA helps DNA code for proteins by messaging the gene from the nucleus out into the rest of the cell using messenger RNA, transcribing the gene into a protein through transfer RNA, and lastly making ribosomes where the transcription can occur through rRNA, ribosomal RNA. And you might ask, why is RNA one strand because it's going to be easier to get the message out of the nucleus messenger RNA and easier to transcribe because it's already missing half of the strand. So you can attach the transfer RNA on to the messenger RNA. So this was just a quick review of Unit 1. Hopefully it was helpful, and we'll see you in Unit 2.